We can finally get to the part of cellular respiration when NADH and FADH2 release their high energy electrons for the production of ATP. There are a couple of places that we really care about when we talk about electron transport and oxidative phosphorylation. The mitochondria, this is a electron micrograph, so using an electron microscope the mitochondria has been analyzed, and I want you to notice the outer and the inner membrane, two phospholipid bilayers, and then in between those two membranes is the intermembrane space. The matrix of the mitochondria is the interior, and then the highly folded inner membrane produce the cristae, which are just a way for the mitochondrion to extend its surface area. The proteins that are involved in electron transport and oxidative phosphorylation are embedded in the membranes of the cristae here. So the mitochondria have adapted to produce lots of space for lots of ATP production. The intermembrane space is also going to be important when we look at ATP production. So let's first summarize what we've done so far. In glycolysis, we made some ATP which could be used by the cell. We also made NADH. In pyruvate oxidation, we made NADH, and we also produced some of our byproduct CO2. So two of the carbons that used to be part of glucose are now in CO2. Acetyl-CoA was shuttled into the Krebs cycle, where we produced 6 NADH and 2 FADH2, in addition to the 2 ATP that can be used. Both of these forms of ATP, or both of these groups of ATP, were formed by substrate level phosphorylation. The CO2 that was produced in the Krebs cycle represents the final oxidation of the carbons that used to be in glucose. And sure enough, a 6-carbon glucose produced 1, 2, 3, 4, 5, 6 molecules of CO2. Now, the NADH and FADH2, all from one molecule of glucose, are going to be used to make ATP in the electron transport chain. So I like this metaphor slide, the metaphor for cellular respiration. In cellular respiration, we put in one molecule of glucose. Glycolysis produced 2 tokens of energy, 2 molecules of ATP. And some high energy electrons were also siphoned off of glucose, and they're being stored to be put into the electron transport chain. Those high energy electrons are in the form of NADH. Pyruvate oxidation led to more NADH, more high energy electrons that are being stored. And then the Krebs cycle produced the most NADH and FADH2, while still producing a couple of molecules of ATP. At the end of the electron transport chain, where all of these high energy electrons get dumped off, we're going to get many molecules of ATP. For every 1 NADH that goes into electron transport, 3 molecules of ATP are produced. For every 1 molecule of FADH2, 2 ATP are produced. And we're going to have this final number of ATP, which can vary, but we'll get to that in a little bit. This final number produced by the dropping off of all these high energy electrons, and then the regeneration of NAD+ and FAD+. The electron transport chain is better studied in a really general form. In the inner membrane of the mitochondrion, there are a series of proteins called the electron transport chain. Within each one of these protein complexes, there are electron carrier molecules. And I want you to imagine that every time an electron moves from one carrier molecule to another, it's losing some of its energy. So it's becoming part of a covalent bond, but within that covalent bond, the energy that it's holding on to is just a little bit lower as it moves down the chain. So I like that this shows it as like a downhill motion. Finally, the electrons are going to be stolen by oxygen. And oxygen is the biggest electron hog, as we know, and also electrons in a bond with oxygen and something like hydrogen, which is this incredibly polar covalent bond, it's a very low energy state. So all the energy that was associated with electrons has now been stripped away. What's been done with the energy is it's used to pump protons into the intermembrane space, the space between membranes. And this is building up like water behind a dam until finally a gate opens up here in a protein complex called ATP synthase, and these protons rush through like water rushing down, rushing from a dam. And that energy that's released, because this is a buildup of potential energy, the kinetic energy that's released is used to slam a phosphate group onto ADP, because remember, it's hard to get phosphates to hold on to each other because they're all negatively charged. The reason why there's this concentration gradient, more concentration of H+, less concentration of H+ down here, is because oxygen is constantly not only taking up electrons but also taking up protons to make water, the byproduct of cellular respiration, a byproduct. As these get used to make water, the concentration of protons lowers, so we can always have this nice concentration gradient being built up. High concentration of protons up here, low concentration down here, so that when the gate opens, there's a rush down the concentration gradient, facilitated diffusion, driving the synthesis of ATP. Summarized here, what I've just described for you. But one thing that I haven't pointed out yet in this picture is that the electrons are dropped off by NADH and FADH2 into this transport chain. So what's happening is a redox. NAD plus is regenerated, which means it's getting oxidized. This molecule initially is getting reduced. FADH2 puts its electrons in a little bit later in the chain, which is why we only get 2 ATP for every FADH2 versus 3 for NADH. But the big picture is these guys drop off their electrons. This gets reduced. There's a series of reduction and oxidation as the electrons move through the transport chain, all the while losing their energy being used to force this buildup of protons in the space. Finally, that energy is used to drive the synthesis of ATP by ATP synthase. If we don't supply ourselves with oxygen, oxygen cannot be used to make water. We don't get a decrease in H+. This buildup doesn't result in the same kind of concentration gradient. If these never get used up, you're not going to have a difference in concentration across the membrane, and ATP synthesis shuts down. That's why we need oxygen in cellular respiration. Your book shows a little more detail. The electron carriers that are found in the membrane, the only thing to add with this picture is that these carriers shuttle the electrons from one protein complex to another. These are lipid molecules embedded in the membrane. I just really care that you know that these electrons are losing energy as they travel down the series of carriers until they're at their lowest energy state when they're joined with oxygen and protons to make water. And of course that NADH and FADH2 are dropping off electrons at the transport chain. Remember that this is the matrix, this is the intermembrane space, this is the inner membrane of the mitochondrion, and this is the outer membrane, which isn't very well depicted here. The process by making ATP, by which we make ATP here, is called oxidative phosphorylation. It's not substrate level phosphorylation, even though this is technically a substrate. It's oxidative because it requires oxygen and because it's a result of a series of redox reactions that cause this buildup of potential energy. So the ATP that's made by ATP synthase and electron transport is oxidative phosphorylation. Let's take a little closer look at ATP synthase. This is then the complex right here. It's a really cool molecule where the rotor grabs these protons, and as protons are rushing through, the rotor spins. The spinning action then affects what we call the catalytic knob. And the catalytic knob is where ADP and phosphate bind and are put together because of the kinetic energy of the spinning of this rotor affecting the knob. So this is the idea of slamming phosphate onto ADP. As soon as this rotates, these protons rotate through once, they're dropped through and used to make water. There are different poisons: rat poison, cyanide, carbon monoxide. These types of things affect the electron transport chain. If something like rotenone affects transport of electrons from the first molecule to the next, you're not going to get the release of energy, and you're not going to get the buildup. Same with cyanide and carbon monoxide. They prevent the export of this electron from the last molecule of the chain to oxygen. And so what happens is we get this buildup of H+, a lack of flow of H+ from the intermembrane space to ATP, and every cell needs a constant supply of ATP, or everything shuts down, and that's lethal. There are some molecules and poisons that can affect ATP synthase itself. They can block the channel through which the protons flow through, or they can also block the spinning of the rotor. But many poisons affect electron transport because it's a very quick way to shut down ATP production, and so shut down the production or the survival of the cell. So to summarize, we have 2 ATP from glycolysis, 2 ATP from the citric acid cycle, and then about 28 total ATP from the input of NADH, FADH2. What we get for every 1 molecule of glucose is about 32 ATP, each one with its 7.4 kilocal per mole of energy to be used in the cell. That's just 1 molecule of glucose. Now, there are other places where we get energy. We don't sit around eating glucose all day. We can get energy from other carbohydrates and from fats, not so much from proteins, and definitely not from nucleic acids that we take in, but we will cover how we get energy from other food sources in a little bit. Before we do that, I want to lecture on what happens in certain types of cells when there is no oxygen. For example, muscle cells. Muscle cells use ATP very quickly because it takes a lot of energy to contract muscles. And even though we can supply our muscles with oxygen, we can't necessarily provide our muscle cells with enough oxygen to produce enough ATP to survive or to continue contracting. This would be particularly important in a heart muscle. So muscle cells, and also cells of organisms that can live without oxygen, have a way to continually produce ATP but not require oxygen. Now, most of our cells can't do that, so we know we need oxygen. But muscle cells can sustain ATP production in conditions of low oxygen, and that is called the process of fermentation, and that's what we're going to talk about next.